Okay, it looks like we are live today, guys. I know a lot of people are excited for this. I'm extremely excited for this. As we know, naturalistic evolution says all things came from a single cell, and that single cell ultimately came from something like a primordial soup. Is this origin of life from non-living chemicals scientifically plausible? That's the question. That is what we are here today to discuss, and I am extremely excited for it. We have got a very special guest. Dr. Ryan Hayes is here with us today to discuss this incredibly important topic. So I want to thank him for giving us his time, for allowing to make this possible. So Dr. Hayes, thank you for joining us today.
Hey, it's great to be here. Definitely been a big topic on my heart and mind for many years. So I'm happy to be here.
Awesome. We are extremely excited. We've been looking forward to this. Before we get an introduction from yourself, Dr. Hayes, I'm going to hand it over to Tony, who helped make this possible. So I want to thank you, Tony, for doing that for us, brother. A few words from yourself.
Yeah, thank both of you guys, Dr. Hayes, SFT. I appreciate it. We're going to get into some of the origin of life problems. It's going to be great.
Awesome.
Okay, well, why don't we hand it over to Dr. Hayes, since this is your first time on the channel. I've got a description of yourself in the video, so people are kind of aware of who you are and what you do. But if you wanted to kind of break the ice, give us a brief introduction.
Happy to. Yeah. So right now, currently, I'm a faculty member at Andrews University in Berrien Springs, Michigan. We're a Christian school here, but I haven't always been a teacher. I actually have worked in the chemical industry for a number of years doing analytical work. I went to graduate school at Northwestern University in northern part of Chicago. And my graduate work was in kind of like a little bit of nanotech, molecular electronics. That's where I came across James Tour, who you guys had on your channel recently. I definitely got into a lot of organic synthesis, learning the rules, learning the procedures, get my hands dirty. And then got a job in the chemical industry doing research, developing new materials, new analytical techniques, and ended up working at a nanotech company in Michigan here for about 5 years as the director of business development. I helped the analytical group just learning how nanotech works and chemistry. So did that to about 2009. And for about the last 12 years, I've been teaching. And I would say really in the last 8 years or so, started just kind of getting on my heart and mind about, why am I here as a chemist? And, what is it about, origins of life and how are we here on this planet? How does life work? And honestly, I haven't been a big fan of biology over these years because I didn't think they were that rigorous, and just fell in love with chemistry. But biology is very important. It's a big field. My wife's a biologist, I should say. And so we often tease each other about our respective disciplines, but just loving seeing how biology and chemistry interact with each other, and just doing a ton of reading on this topic here in the last 8 years or so. And full disclosure, I go out and give talks on these areas. I give talks to my students. I've been out to camps and churches and things within our church here, community. I'm a Seventh-day Adventist Christian, and I love this topic. It's very interesting to me. I think about it all the time, so it's great to be here. And I'll maybe turn it back over to Tony. May have a few kickoff questions, and we can get going.
Absolutely, sure.
I was going to. Oh, yeah.
Go ahead, brother.
I was just going to say that's an awesome introduction there, Dr. Hayes. Clearly, you're more than qualified to discuss this topic. I agree. It's an extremely important topic, and we are honored to have you on with us today. So I just wanted to thank you again for giving us your time. So, Tony, if you wanted to kick us off with some questions, go ahead, brother.
Sure. We'll get into some of the origin of life scenarios. The most current ones, probably protocells or RNA world, metabolism first. And to go all the way back to the most famous Miller experiment, if you wanted to kick it off with that one and some of the problems that they had.
Yeah, that's a great one to get started with, the Miller-Urey experiment. And, it's amazing how that particular experiment really persists, I think, in the hearts and minds of a lot of scientists. And that's an old experiment. And was it reproducible? Absolutely. That's a reproducible experiment. they were able to generate amino acids from some kind of primordial soup, electricity, and some raw ingredients in the atmosphere, and reproducible. And that's very interesting. They form some really interesting, amino acids. And and everyone gets excited about that. And that was very exciting. But how much of it has it progressed since then? Not a whole lot. That experiment, there hasn't been really, there's maybe a few adjustments to it over the years. And I think even a lot of evolutionists would look at that and say, yeah, that experiment had a lot of flaws. The gases they used maybe weren't the right ones. They got sort of a tarry mix, and the amino acids that were formed, there wasn't really any peptides where the amino acids were hooked together.
They were also racemic. They were totally racemic and useless. They didn't go on to produce proteins or anything functional.
There wasn't anything functional of that. You've got a few interesting molecules in there. And I think we all know now that life is not a few interesting molecules that have come together. It's a lot more than that. We see a lot more organization and things. And my biochemistry colleague here is often reminding me of how hard it is to get amino acids to come together. And this is why the theory of amino acids coming together, for the formation of life has been discarded. You can't. Yeah. It is so hard. Why is that so hard? These little amino acids. Why can't they should polymerize and make all sorts of fun things? Well, and we know why. The equilibrium constants, the equilibrium reaction that are there. Once it's in water, it favors the breaking apart of the amino acids of the protein, the polymer, back down the amino acids. So you have to come up with a really clever way to overcome, amino acids, the breaking apart. And that's exactly what we see inside of an actual working cell, is a really clever set of, machines that come together to overcome the poor chemistry conditions to bring them together. There needs to be
I don't know of any serious chemist that even uses those spark experiments anymore. They just, like you said, they're just obsolete chemistry, flask experiments.
No, it was interesting. Those are fun experiments.
Right.
And that was great that someone tried that, and fine. But I mean, honestly, it hasn't really. Why does that one stick in the textbooks? There's a lot of textbooks that still talk about that.
Yeah. Yeah, I mean, it's pretty much well known as a failure, but a big atheist talking point as being such a huge success still.
Right, I hear about it all the time still.
Yeah, amazing.
Yeah, and that's the thing. There's a lot of these legacy things that hang on, and I know guys at the Discovery Institute, they've written books on this. They call it zombie science. These guys, they just keep resurrecting these dead scientific arguments and bringing them back. And so, what are the modern things? And clearly there's modern day research that's going on, but why aren't those making it into the textbooks? Why is this dead science from the Miller-Urey experiment still hanging on there?
We could fast forward 50 years later to the RNA world. I think it was in 1986, Walter Gilbert first proposed that. But then again, we have a lot of unstable scenarios with this RNA world, RNA peptide world. They seem to come out with a new RNA world every 5 years because it's just right with so many problems.
Yeah, that's always put on my slides. What RNA world version are we on? And I understand you got to throw out hypothesis and come up with it. I think it's important for people to understand that there's millions of dollars, a couple $1 million thrown at a number of scientists every year through NASA, the National Science Foundations, that are looking at the chemical evolution of life. And if you want to keep that money, you have to publish. And you're going to spin it as broadly as you can and as exciting as you can and get the best titles you can. And they keep chipping away at really tiny things and never really, I think, advancing the chemistry. So you can't have DNA first. You can't have proteins first. RNA, I mean, that's a real stretch to use RNA. It's got to be the catalyst, the information carrier. Right. I'm sure you guys have talked about the information problem.
And then there's always a pre-RNA world. There's always a pre-ribosome world or a pre. They're always going before something,
They just move the goalposts, move them back, and that's just not what we see. when it comes down to a lot of chemistry, which is why I'm here and why I think we need to be saying a few more things. The chemistry of these molecules is it's hard to bring these things together.
Right. They're never in the right structures, desired structures that we need for life.
Exactly. And so you guys had some great discussions with Dr. Tour, and I think there's a few other things to start adding into this problem. And one of the things that a couple of different things that I like to talk about. And early on, if there's going to be life on this world, of course, it probably started around thermal events, if you believe in evolution. And the theory is, maybe around a thermal vent, but not too long thereafter, photosynthetic organisms started to oxygenate the planet. Okay, sounds easy enough. Has anyone ever looked at photosynthesis? And absolutely, we understand photosynthesis very well. And this is a very complicated set of molecules and system that's inside of a plant. And this is a little bit more my graduate work was involved with. I looked at light capturing molecules and how to, you get them excited with certain colors of light, and then how they move electrons around. Some of the basic steps of photosynthesis. And photosynthesis had to be one of the first things. If life was going to get evolved, that had to be one of the first things. Because how do you oxygenate a planet? Because it turns out, as scientists, there's no way, no quote unquote natural way to oxygenate a planet. How do you even oxygenate a planet so that you could have an oxygen living system? You can't heat rocks, boil water. There's not a good way. So the books I've read, the research I'm reading says the only way to oxygenate a planet, living systems. You need a living system.
Yeah. That brings me to another atheist talking point. Well, microenvironments. We're going to find the just right environment with all the correct molecules, the just right amount of UV radiation. We're going to have all these selection processes happening in some chemical puddle under a rock somewhere. I mean, it just seems really far-fetched at that point.
Very much so. And I think the other things that are forgotten at that time, if you're early on, the radioactivity on the Earth was much higher. If you dial it back, you look at where we are now, it had to be more radioactive. That's so destructive on DNA and RNA and proteins, ripping things apart. So you got to be fighting against that. Obviously, all these things you got to get the concentrations right. What molecules are even making? They could be all over the world, and trying to get them all in the same spot. And so I would go back. So what I learned from this is my personal experience of doing chemistry. If you want to absorb light and move electrons around, those systems, those molecules have to be positioned exactly in the right place, or the electron transfer events to create energy, to do things, won't happen. They can be off by nanometers, less than that, picometers, and you just won't get the right electron transfer rates, and your whole system falls apart. And I built things that had like 3 molecules in them. And I struggled to put 3. I got a few papers out of that. We did some really interesting things with molecular electronics. And people who are trying to put together artificial photosynthetic reaction centers, that's a ton of work. And they're basically co-opting this. Let's just take this part of the plant and put it in this part, and we'll kind of tweak the plant a little bit and some of the systems in there. Inside photosystem 1 and 2, there's hundreds of these very light absorbing and electron favorable molecules. And if there are arrangements of these, not 3 like I did in my lab, 100 or hundreds of them, and if their positions are off just by a little bit, photosynthesis doesn't work. And that complicated molecule and system had to be one of the first things on this planet. The chemistry is not supporting that at all.
Right. A lot of wishful thinking, I think, by the atheist camp.
Yeah, it's wishful thinking because that's not what the science is. And if they're asking us to be scientific and asking, Christians and other, hey, be science. Here's science. Here's what science is showing us. I'm following what I know from being a chemist, being a scientist, what I read, what I understand. And how in the world are you going to get something as quote unquote simple as photosynthesis to happen? The whole origin of life, it's dead in the water. I don't know how you make that. How you make life.
Right. Give us an idea as a chemist, some of these purification processes, because James Tour talks about this. You've got to purify at every step of your experiment. So how are we getting all these 100% pure chemical reagents on the early Earth or anywhere?
No. Yeah. So my advisor in graduate school, his favorite saying was garbage in, garbage out. Garbage in, garbage out. It was so. The reactions I was dealing with at the time, we had about a 2% yield. But my important molecules were in there. So what did I do? I had to hunt around. I did so many separations. Thankfully, they were very beautifully colored. So I could go using my eyes and go, I need that one right there. And I could sort it out.
So interference, always interference by the chemist, by the hand of the intelligent chemist.
You know, at some point, we've got to stand up and recognize that all of this is, hey, anything that we're making work in a useful way is involving intelligence. There's a mind in going, this is not where the chemistry is going. Okay, but if I move this part with this part and I rearrange them out of their quote unquote natural order, we make things happen. We're making it happen all the time.
Absolutely. James Tour is famous of saying, well, we'll just give them everything they want. All the activated nucleotides, all the left-handed amino acids, all the right-handed sugars. Okay, yeah. We'll put it in the perfect lipid membrane in a protected laboratory environment, piece it all together and produce this primitive cell. Do it.
Do it. Do it. I would love to see it. I encourage that research of building protocells, understanding what the simplest cell looks like. It still looks like it needs hundreds of genes still. We're talking about thousands. You know, I try to question Tour on this. Hey, how many molecules do you need for life?
Right.
I think it like blew his mind. Is like, what do you mean? There's so many different ways. It's so many different molecules. So I've asked a few people, and they said, you know, maybe the simplest life, we may need 3,000, 4,000, maybe 5,000 chemicals.
Wow.
You know, so my analogy is, hey, go make some cookies. That's actual chemistry right there.
Yeah.
Sure uses the car analogy. I like the car analogy. I think that
cars went. Yeah.
But go in the kitchen. How many ingredients does a cookie need?
And all at the right steps and isolated and added at just the right time. And it's got, you know, of course.
You can't
manipulation.
You can't just throw heat on the ingredients and hope for the best. Nobody does that. You don't. You can't. There's a mixing problem. You may have everything in there, but if you don't mix it right, I've lost my batch of cookies that way. I've lost my batch of chemicals that way just from mixing.
Well, that's actually another great point. Let's go to the famous time argument. We're not giving these chemicals enough time, but every chemist knows you can't leave your reactions overnight. You can't just leave a bunch of test tubes sitting there with all your chemicals and amino acids.
Correct. Your molecules degrade. They're not infinitely stable. So they have a shelf life. Everything has a shelf life. There's side products that are constantly being made. You know, and let's face it, the most of those molecules that you need for a living system, they need a living system to make them. This is so chicken and egg, it's ridiculous.
Ridiculous.
So the science is not, hey, the more we study how a cell works, the more we study how bacteria work, love it. Let's see it. Let's see all the complexity there. Let's see all the parts coming together. Try to make life. I'm all for it. Try to make a protocell. Let's see what are the big, because that is helping to communicate the design, that the parts that all had to come together at the same time.
What about Craig Venter? About 10 years ago, he claimed to the artificial life in the lab. But then when you got into the details, after you read some of the articles, about 6 months later, some of the science, even secular chemists were questioning what he did.
Yeah.
And then you get into these details. It was, I had to use a pre-existing genome from a goat cell. I had to purify a bunch of reagents. I had to, select the right exact pH. I had to splice in some DNA. I used a pre-existing membrane.
Yeah. Yeah, right. You come on. When you borrow all the parts from some of that, I mean that's quite a stretch, even for that's a stretch for most people once you start looking into that. So very interesting work. I think it just underscores how many different parts that are needed to make life, and so to stretch that and to think that you've made life, that's being a little bit honest there.
Well, one thing
exaggerated.
One thing that I find interesting, I was just going to point out based on what you're saying, this is an awesome discussion. I love it. But even the fact that life itself isn't just based on biochemicals. You could have all the biochemicals you want, like Tony was saying earlier, and it's still not going to give you life. You can have all the amino acids you want, the proteins. Heck, you can have RNA, throw in a little DNA. But number one, they will never assemble themselves. And even if they did, we still have not introduced information into those molecules. Isn't that right, Dr. Hayes?
Absolutely. The information argument for me is huge. To me, that's a game-changing argument. That is something I don't think evolutionists have, because they can't. I honestly, that's a deal breaker for them. They can't address that. How do you get the right sequence? This is so obvious, even to a non-scientist, that, hey, you're right. Once you understand that there's a code inside of you, and that code, that information has to be just right, and there's more ways to get the code wrong, almost an infinite number of ways to go wrong than to go right. I think this argument is substantial. How do you get the information? You can have all the molecules in the right place. You can have we'll give you all the DNA, we'll give you all the RNA, all the nucleobases that you want. Okay, which order are they supposed to go in?
Right.
Yeah.
Just too many ways to go wrong. If you
all in chiral form as well.
Yeah. We haven't even talked about chirality.
We haven't got into that yet.
It's almost impossible to get there. Yet the more we understand about science and chemistry and the chemicals and how they all work, we're understanding the beauty of what's there, and that, we can do this. We can do this. So another thing I think it's important to think about is thermodynamics and kinetics. But in my general chemistry class, we often talk about entropy and enthalpy and these things. And these sort of arguments have been out there for a long time. And a lot of Christians will have said, well, the second law of thermodynamics, you can't overcome entropy. Well, and then rightfully so, a lot of evolutionists come back and say, well, no, if you put energy in, you can overcome entropy. And this is partially true. But both of those arguments are lacking the actual substantive thinking that actually goes into how chemists look at thermodynamics. You look at both what's called the enthalpy. Are you putting energy in, or does energy come out? Then you look at entropy. Is the system coming together, or is it spreading apart? So I break this down for my students. And I say, look, there's 4 classes of reactions that chemistry falls into. There's this type where doesn't matter what the temperature. You put them together, boom, that chemical reaction happens. We call it spontaneous. I think the world is looking to call it natural. That just naturally happens. Boom. There are 2 other types where if it's hotter, if you have enough energy, it will overcome the entropy, the reaction happens. There's a third set, where if it's cold enough, you can actually you can overcome the entropy that's there, and which is weird sounding, but cold enough and it will work. So these 3 categories are what I call very natural, spontaneous reactions that take place. And then there's this 4th category where it doesn't matter what energy you have, it doesn't matter what the temperature is, you're fighting entropy, you're fighting enthalpy, and the thermodynamics is just totally against you. Totally against you. Now I ask, where do the molecules and the chemical reactions that we need to form life, which one of those 4 categories do they fall into? Are they the spontaneous ones that if you just give enough time, or maybe a little energy, or maybe you got to cool them off, they'll happen? The answer is no. All these life-giving processes, for the most part, fall under this 4th category of they're not spontaneous. They're not naturally occurring. It doesn't matter how much energy you put into it. In time, they won't fall. These reactions do occur. How do they occur? Intervention by intelligence.
In the lab.
You work this part with this part, and we make these non-spontaneous chemical reactions happen all the time. As humans, when we interject our intelligence and we learn tricks to overcome that. But if you leave nature to itself, it's going to fall into, the only things that kind of work are in these first 3 categories. And none of those are not the majority of chemical reactions that life is made of. DNA come together, proteins come together. They all fall into this last very, very unfavorable category. How are you going to make it work? Intelligent.
Yeah, it's funny that you mentioned that, because we're hearing that from some of these origin of life researchers themselves. People like Robert Shapiro, he was known as Dr. No. He would go into these RNA World meetings and just shut everybody down, because he would just point out how this stuff was so unstable. It wasn't going to form anywhere other than in the lab where it's being designed.
Right. We're proving over and over again how do you make life? Intelligence. When you put a chemist in there, you put a scientist in there, you put the baker in the kitchen, you can make all sorts of great things that is not natural. And so my beef that I have with this is it's the wording. People think that if something is natural, that it just occurs. We live in a planet where everything's automated and just so happens, and we're so ingrained with that. I have to teach my students what natural is. What is expected, because they don't know. We live in a planet with all this water. Having water on the surface of a planet is actually a really hard thing to do. It's really hard. And I actually think that maybe our planet is intelligently designed, and I have some evidence to back that up. That's another talk for another time. But we're giving them that. We're giving them a life-permitting universe. We're giving them a life-permitting planet with all the right atmospheric constituents and all the chemicals that possibly you need in Earth. You're overcoming. You have to overcome thermodynamics. You have to overcome the information. And I call them wells. They're deep wells. How do you get out of these wells? And there is not any plausible chemistry to fully get out of the well. Yeah, you may be doing these cute little reactions, but that's only 1/100th or 1/1000th of the challenge that you need to make life. Absolutely. So I get pretty passionate about it as well. And I'm just not seeing it as a chemist. I'm just not seeing it. And why you say, well, why aren't chemists getting out and saying things? That's not what. Look at our classes. I don't have a historical chemistry class. Let's look at this. Or we don't have intelligent design chemistry classes. That's what chemistry is. We're trying to use our intelligence. Chemistry is still kind of a young science. If you think about it, I would say modern chemistry was 30, 40 years at least and longer after Darwin. He had no clue of what modern chemistry was about, no clue what was in a cell. And here, why are we standing up on this, for this guy who didn't know anything about chemistry or anything about modern science? We're pushing a dead theory around.
Crazy. He rode a horse to work, and we're still listening to him.
Yeah, that is so. And obviously, evolutionists are looking for evolution 2.0 or 3.0 or 5.0 or whatever version they're on right now, and they need something, because all we see over and over again, repeated. When you put intelligence into the system, you can overcome the thermodynamic challenges, the purity, the chirality, challenge, and we do it. Now, we don't have the expertise to go, I don't think at this point, to start with raw materials and make life, but we understand the parts. We're getting a pretty good understanding what parts are at least involved. How do you pull them together? I don't really think. So I think the big bet is in 50 years or 100 years, some magical theory is going to show up, and and they're praying to this future theory that it will come and save them. It won't be there.
Well, one thing I found really interesting with what you're saying is I do believe the information problem is destructive to them. And the way I think about it, Dr. Hayes, is I think about it like information in a book. The ink molecules itself is not the information. It is ink molecules written on paper. That's it. You could pour ink on a page, for example, and you would not be getting information out of it. The information, where does it come from? It comes from a mind that has actually organized those molecules into words, sentences, paragraphs, and chapters.
Absolutely. What opened my mind, I read Stephen Meyer's book on the signature in the cell. And I don't know why I hadn't thought of it before, but we do need help from other scientists to open our minds and go, hey, I never thought of that before. And he was talking about the DNA. And I thought, from my mind, I thought, DNA probably just organizes itself into ways that make informationally rich structures or something. And then I started looking at it and said, there is nothing about, if I cover up the nucleobases and I look at the ring structure, or the backbone, the ladder that holds everything together, there is no driving chemistry to organize it. If there was, we would see patterns. It would be, A, B, or AT, or AATT, or even some complicated complex. And no, it is totally scrambled, but yet it's useful. It's this very complex but very specified pattern of information that we see. It's exactly represented by computer code and by things that we write.
Dr. Hayes, can you give us an idea of some of the designed equipment that they're using? I mean, DNA, RNA sequencing machines, all kinds of equipment in the lab, state-of-the-art stuff. Lee Cronin, for example, uses the latest equipment. I mean, this stuff is any relevance to prebiotic chemistry.
Absolutely. In my graduate work, my job was to build these molecules to capture light, move electrons around. We actually partnered with a group that they had the DNA synthesizer. So some other of my research was dealing with trying to use DNA as an electrical wire. And so I got to learn about the DNA structure, and they had their DNA synthesizer turning out little micrograms of DNA. Why? Well, because you can automate that when you know how to, you put your intelligence in it. We know the process. You can build these things. But absolutely, advanced chemical machines, little factories. But you know what, Tony? That's exactly what we see in the cell. Little fancy machines, the highest technology, not in our labs, the highest technology that we hope to attain as scientists is sitting right in a cell. That is high tech. High tech.
The answer is right under their nose. They just don't like it.
Yep. They don't like it. And it's actually changed the way I look at bugs and mosquitoes and stuff. Because what are scientists trying to do? We're trying to build little nano robots. Dude, we got nano robots flying around us all the time.
Yeah.
that self-replicate and feed themselves. Yeah. I mean, high tech.
But they're not really designed though. We just got to ignore all the design in them.
Okay.
It's not really there,
So why are we trying to mimic this stuff?
Right, exactly.
Awesome. Because it's awesome. Biomimicry. It's like, well, give it billions of years, it'll work out. Yeah, that's the lie. That is a lie. Billions of years will get you a pile of sand.
Yeah.
That's it.
Well, I find it amazing that just listening to this amazing conversation, I've been writing down so many things, but it seems like the most common scenario for the origin of life right now is the number of RNA world hypotheses like you gentlemen have been talking about. But the way I understand that is they are imagining an ocean or a lake full of RNA molecules that have then learned to reproduce themselves and then slowly somehow evolve into the first reproducing organism. But Dr. Hayes, you were talking about how water tends to drive the reaction in the wrong direction. And a primordial soup, wouldn't it have lots of water in it? So how is this even remotely plausible, Dr. Hayes?
Well, the hypothesis then is that these things occur on the edges of the warm little pond. And so where it dries out, and if you have the right rocks that can catalyze it there, once again, a very designed system, it may happen. But, these things are only generating, these ideas are only generating a small fraction. Even if let's say it did work, it's only generating a small fraction of the needed materials to create a living system. It's not enough just to have RNA. When I look at the numbers, this is what's so amazing. I did a talk actually this weekend on how our cells are programmed to die, apoptosis. Why? They're messing up all the time. Every day, 50 to 80 billion of our cells have fallen apart, and they need to be degraded and cleaned up. And every day, thousands of mistakes happen in our DNA. Why? Because of the radioactive planet that we live on, DNA is being shredded. But somebody thought about that, and there is these amazing DNA repair mechanisms going on in our body. So it isn't just enough to have the molecules. You got to be able to fix them on the fly because they're constantly breaking down. And somebody thought of that and put that system in, was it 2015, 3 groups of chemists got the Nobel Prize for the DNA repair mechanisms that we find in our body. Not in our body, in E. coli. E. coli, one of the simplest bacteria, have to have the DNA repair mechanism in it, or all of its DNA and RNA are going to turn into shreds within a couple of days.
Dr. Hayes, I got to mention the RNA world on clay. This has been talked about a lot. But basically from what I'm hearing is that the clay is chemically stabilized, and they use all these assisting substances. And
right.
So, I don't know how that is going to happen anywhere.
Okay, good. You make it. But that stuff's not going to sit around for the thousands of years that you need it to eventually hook up with something else to get into the right soap bubble that starts to act like a cell. And if anybody thinks that a soap bubble and a cell are equivalent, they're greatly mistaken. Greatly mistaken.
What they're calling life is just absurd. It doesn't reproduce. It doesn't metabolize. They're just calling it life. Like what James Tour was saying when he asked these chemists, "What do you think life is?" "Oh, just a little bit of RNA in a membrane."
No.
What?
Well, because that's what their great leader Darwin was saying. It's just a few of these molecules, and And that's you can twist the facts all you want, but there's thousands of different chemicals that have to come together all at one time to have life. And at least there may be something else, the spark of life that we don't even know about. But at least from a chemical perspective, this is a cookie that I cannot bake in my kitchen, or chemists can make in their lab. Hey, if you're borrowing things, if you're borrowing ATP synthase, that's what Venter did. He borrowed some of these things. Yeah, sure, you can make it, but that's cheating, man. Get your own dirt, I think the joke goes. Get your own dirt.
Well, that's the thing. These chemists are purchasing all these synthetic kits. They've got all these molecules pre-prepared before they start their experiments.
Well, this is what's a bit challenging as I deal with my biology friends. They're all biological. They have a hard time even making simple solutions. And hey, can you come make this solution for me? And it's like, this is not that hard. You just take a little bit of this and this and add water. Well, could you help me with that? And there's a great misunderstanding of how chemistry works, and it must work out because when I buy it, it's there. You know, I purchased it. No, there's a great amount of intelligence that went in to make it easy for the biologist to put things. So they're kind of at the end of the intelligent design process of giving them chemicals, and so I can appreciate how they may just think, hey, this stuff comes together. Get in the lab, you don't have to make life. You don't have to make life. Just go make me a porphyrin. Go make me a little hemoglobin. Go make me just something simple. Go make me a protein. There you go. Go make me not even protein. Go make me a polypeptide. Go make that and come back and talk to me in a few years when you have failed and had a really hard time, done all the cleanup, done all the, you got to do all the analysis. Okay, life doesn't have to do the analysis, just has to figure out a way through it. But I mean, you're going to get lost pretty quickly with all that messy chemistry that's going on. Garbage in, garbage out.
Well, one thing that I wanted to branch off, because it's something you said, Dr. Hayes, I found fascinating with the DNA repair mechanisms. And now that they're looking to this RNA world, well, RNA, as we know, and correct me if I'm wrong, is more unstable than DNA. And as you were saying, in our cells, there are about, I believe, 1 million DNA breaks every single day that are, most of them are fixed by this amazing all these amazing repair systems in the cell. So my question would be, if RNA is less stable than DNA, and DNA requires many amazing DNA repair systems in order to survive over time, how in the world can an RNA world then be plausible? Like, what would
it's not. It makes me wonder: is there RNA error correction in there? And I think so. So to kind of build on that, how do you know what to correct it to? Just because you have a correction system, how do you know what to correct it to? Anything that gets fixed in our world, you have to have a knowledge of what it was originally, and you have to have a special tool in order to fix that. If you're correcting something on a piece of paper, you have to have whiteout, right? And if you're going to correct a computer code, you can't use whiteout. You have to use something else. You have to get in actually and know what the code is and program. Or if something on your house breaks, you're going to use a cement tool. If you have a stucco coated house like ours, don't buy a stucco coated house in the north, by the way. Terrible chemistry. But you have to have the right tools to go there and fix it. So this stuff is breaking down all the time, and I think we're all thinking this glorified world of, hey, we put a few chemicals together, they're going to come together, they're going to work eventually. Yeah, because that's the world. You live in an automated world because of the hard work of a lot of chemists doing the purification, doing the thought process for you, all the intelligent design that's gone into the process. And let me tell you, if you see automation, there's a supervisor and some intelligent behind the system, behind the scenes working the system.
Yeah, so it's all strict protocols and procedures. And like you said, the biologists really don't know anything.
Now, when it comes to chemistry, hey, my hat's off to all of the interesting connections that they've made between RNA and DNA. But when it comes to making things in the lab, they've taken general chemistry and organic chemistry. That just gets your foot in the lab. It's the years in the fume hood and cutting your teeth, making failure after failure after failure, and learning this stuff just does not come together. That's what science is showing us, not the opposite.
Excellent.
Yeah, I mean, that's why we have to write these papers and prove people, did you really make that? Yeah, here's my proof. Boy, because they don't, that's really hard to make. Yeah, it's really hard to make. And here's my proof, here's my steps. And then there's the whole reproducibility issue going on in science right now. I don't know if you guys are aware of that. There's so much work is not reproducible.
Right, but it's passed off as in their papers, and just believe it. Just believe it because some biologist said it.
Yeah, and nobody sees the retractions. Nobody.
Well,
sees
Jack Szostak retracted his paper. I believe it was 2 years ago on self-replicating RNA because he couldn't get it to work under prebiotic conditions. He did it in the lab, of course, under very highly controlled conditions. Okay, sure, I agree with that. But would that work anywhere out in nature ever? The short answer is no.
No. And so that's the dishonesty that I really feel that's going on. We've got work that's not reproducible. You get one thing that sounds plausible. That's what sticks in the textbook. That's what sticks in the news. That's what sticks on the Google searches. And we're not seeing the full picture there. And honestly, it's taken a long time for me to sort through it all and to go, wait a second. A lot of these things that are held out as these awesome truths to support evolution and the origin of life, man, those are just paper mache things that are just facades. And the truth is way behind, is nothing of that. And a lot of those experiments were junk. But, a lot of our biology faculty don't even know that.
Right, exactly.
They don't know that, oh, wait, that didn't work? That didn't work? No, that was disproven. Really? Yeah, I mean, even evolutionists don't even believe that anymore. Oh, we probably shouldn't be teaching that anymore. Yeah, you think? So, and this is at our Christian schools and things. They're caught up in what the textbooks say. And yeah, it's funny in the biochemistry textbook, they used to have a pretty big chapter on the origins of life and how the molecules somehow figured out how to replicate themselves. It was such a story. It was incredible. I'm like, are you kidding me? And then a reference to all of the false science and said, well, this stuff's probably not right. Then why are you putting it in a textbook if it's probably not right? That was in the caption. This is probably not correct, but they leave it in there. That was the whole Haeckel's embryo stuff. Now they leave that in there. They draw some pictures, and that section in the biochemistry textbook is getting smaller and smaller and smaller. I think actually Michael Behe wrote about this in Darwin's Black Box. Well, if this stuff is true, shouldn't that be becoming its own chapter? Maybe it's a textbook, and yet it's getting smaller and smaller, and they're taking this stuff out of the college textbooks and just have a little bit of here and there. Why? Because it's not holding up scientifically.
I think they're running out of scenarios. I don't know where the RNA world is going to go next. The moon? I don't know where they're going to put it.
No, they're already saying where it's coming from. Aliens brought the stuff here.
Oh, okay.
Directed panspermia. Is that what that is?
Yeah. Hey, that's becoming one of the leading. I don't know. It's becoming one that I'm hearing. I'm like, really? You just passed us off to a bunch of aliens? Well, who made the aliens, right? And anyway.
Exactly. It just pushes the problem back even further.
Yeah, exactly. And I know they put it back on, well, you guys are just God of the gaps, and God came in, and God did that, God did that. Well, because that's scientifically what I see is that you need intelligence to come in and put these parts together. We're trying to put the parts together. We're trying to see how they come together, and they don't. And so you need some kind of master thought, a chemist in there going, okay, I need that, and I bring that in, bring this in, bring that part, and that's not a natural part, so we got to have this made and all these. Okay, turn the key, start, go. And if you try to do it piecemeal, who builds a card piecemeal? Who builds anything piecemeal? We have plenty of examples around us. Why can't that stand up as like, well, they're that factory? You don't build a factory, you bring in all the pieces and then go. You get the system going when all the parts are there. Cars, watches, you name it. A loaf of bread and cookies. You bring all the parts together and then you make it under very controlled conditions. We're not seeing that from the origin life. We're not seeing that from evolutionists. We're just getting a bunch of hand waving and very large titles that are overreaching. And
I wish we had more outspoken chemists like you and James Torr. Excellent.
Well, and part of it is we don't have these classes. We don't have the training. I had to get it myself. I had to read it for myself. I had to think about it for myself. A lot of late nights, a lot of book reading, and then thinking about it. It's just, we're too busy, honestly. As chemists, we're too busy. We're analyzing things. We're synthesizing new molecules. You're trying to fight disease. You're trying to fight, you're trying to make people healthier, or some chemists are trying to make them fatter. I don't know. You've got analytical work, synthesis work. There's just a ton of other things. We don't have classes where you sit around and think of this stuff. Why? Because we're too busy. We don't need it. It's not critical. So this is just, honestly, it's not something chemists have any lectures or classes in, unless you come to my school, and then you'll get some on the side. There's not an official class yet that we have, but I give some bonus points for my students to come hear some of my lectures on this, and it helps them to learn chemistry.
Dr. Higgs,
that's why interviews like this are so important. And I think that's why I know I'm personally getting so much out of it. And based on what you were saying on how the chapters in the textbook seem to be getting smaller and smaller, is what the naturalist or materialist, what they base their belief on when it comes to abiogenesis or chemical evolution. It seems like they're not basing it on empirical science that's actually demonstrable, repeatable, observable.
No.
So what are they basing it on? Just blind faith?
A hypothesis, right? It's all that part about science, right? Make a hypothesis. Okay, well, let's just keep making a new hypothesis.
Right.
Keep that going.
Would you say this is the most dishonest field of science right now? Or
that's a good question. Certainly way up there. There's maybe not most, but it's got to be right up there. Mainly because there's a lot of money in doing cancer research and all these other things. So as anybody know, pharmaceuticals, big money, and not that they're lying or cheating or anything. That's just hard business, and that's where people are going to cut a few corners and make their molecules look good, so they can get some money. So there's a little bit more money in that, but this has got to be right up there. Origins of life. Hey, there's money in it. You can get a little bit of grant money. And it's hard to get grant money. But if you can spin a good story, I mean, honestly, I've just heard it from scientists who say, this doesn't support the materialistic view, but, I can get funding, so I'm going to make it try to fit. So I honestly heard that from a few scientists. That's just so dishonest to me. It's very bothersome. It keeps me up at night, honestly. My poor wife has to hear it late at night. I'm just, what? Here we go again. Kids are sleeping.
Well, when you've heard it a million times. And one thing, Dr. Hayes, because I'm writing things down like crazy because so many good points and topics. One thing that came to mind when you were talking about the chicken and egg problems that abound in the origin of life scenarios, from my understanding, enzymes are obviously needed as a catalyst in the cell, and RNA can do this, I believe, in a very limited extent.
Right.
But the reality, and correct me if I'm wrong, is that in living cells, protein-based enzymes do most of the work. Therefore, I'm wondering, how did life change from RNA enzymes, from which there is only limited support, to this amazing protein, these amazing protein enzymes? When did they say this occurred? I mean, it just seems like a lot of storytelling, like you're saying.
Yeah, absolutely. Bait and switch. It comes to mind when you're telling me that. Bait and switch. Oh, let's get us focused on RNA. Okay. What if that works? You got to switch it over to a protein base.
Right.
And I'm not a great biochemist, but I've been looking at a lot more enzymes. Awesome little machines. And we got to start calling the right thing, because we're getting caught up with the words. And so when I give my talks, I feel like I have to teach this. Like, what does natural look like? What's a machine look like? Because we just think it's chemistry. Chemistry's hard. Honestly, chemistry's hard. And who wants to go learn something hard? And so words like natural, like, honestly, we live in a very unnatural world. And that, like, what? Wait, no, nature's natural. Nature, one of the most unnatural things out there. It's all of these unnatural parts. When I mean unnatural, I mean non-spontaneous. These molecules are not spontaneously forming anywhere. So something had to make them and bring them together, automate the production, automate what we call reproduction, so, self-reproducing organisms. And we live in this automated world where things just happen. And so we get this idea that it's easy and that it just happens. And that's not exact, and that's totally the false dichotomy there. So we have to actually I think we have to use different words. We have to use, it's just natural. No, not natural. Not natural, honestly, because if it was, I could just throw it in a test tube and this stuff would come together. Boom, right. We'd be making cells. Not natural. Life. So that's my quote. Life is not natural.
I like it. So then based on what you're saying, I mean, there's chicken and egg problems over and over again. And when it comes to this specific situation, because we have information in the DNA that requires enzymes to read it, but then those instructions to build the enzymes are on the DNA, which cannot be read without the enzymes. What came first, right? The enzymes or the
DNA. Yeah. Yeah. So that I mean, evolutionists and naturalists, the materialists, they threw out the enzyme first world because they knew you need the DNA to make the enzymes, and then you need the enzymes to make the DNA. So that has been debunked, and no one's even going there anymore. That one's been defeated so long, 20, 30 years ago. That's not, because we know you need the factory, and the instructions, and you need all the parts there. So that's been written off a long, long time ago. So this is why, hey, RNA, we don't really know a whole lot about it. Let's go into this little area that we don't have a good view of, and let's put our eggs in that basket.
There's always another chemical pathway hypothesis of, it's going to work.
Yeah.
It's going to work.
Hey, hey, fine. Go ahead. Go make another hypothesis. But eventually we're going to have to use science and go, well, how does this stuff come together? How does it come together? Because it is together. We right now, the leading theory of how things that are very complicated, need purification, how does it come together? Intelligence. That is our leading scientifically validated point. Intelligence. It's proven over and over again. And I know James Tour doesn't want to go there, and I appreciate that. I appreciate that, because if you go there, and I'm going there, you risk publication. Where's the science in that? You risk promotion, which we're a Christian school. They like that I'm talking about this stuff. But you risk your professional livelihood, and that's the other reason why a lot of scientists are not saying anything. They're being strong-armed into being quiet, honestly. So that's not very scientific.
Well, it's such a great point you bring up. How many failed hypotheses does it take to finally conclude the obvious?
When the money runs out.
When the money runs out. Yeah, exactly. Wow, so many good points. And actually, we've had a great audience tonight. I should say, this afternoon, with a lot of good questions. And we often get them in the forms of super chats, Dr. Hayes. For example, we have one here from James Carter. He's given a super chat, and he said, hopefully I don't butcher this, but he says, doesn't Le Chatelier's principle make abiogenesis impossible, since an obvious primordial soup drives the reverse of polymerization hydrolysis? Does that?
Oh, that makes perfect. Yeah, I was trying not to use all the big words there.
Right.
That's a French word, Le Chatelier.
Le Chatelier, there we go. Yeah, I guess I did butcher that.
He's right on. That's what I was trying to explain with the equilibrium. Le Chatelier's principle pushes that. Now, you can try to overcome Le Chatelier's principle if you use intelligence. In fact, I can give you so many examples of where Le Chatelier's principle makes the chemical reaction terrible. In fact, ammonia, by the Haber-Bosch synthesis that was developed in the early 1900s by some German chemists, that thing is so unfavorable. Well, I don't know if it's unfavorable as protein and these things, but using their minds, they were able to shift and drain off the product. There's things you can do to trick Le Chatelier's principle that people using intelligence can overcome Le Chatelier's principle, but you have to fight it. Le Chatelier's principle is fighting you to not make proteins. So using intelligence, you can get around that. It's a great point.
There's always more against it than for it. It seems in all these reactions.
You turn over the next rock, or you learn the next thing about life, and you go, oh man, that's hard to do. And you gotta do this, and you gotta do that, and you gotta fight this, you gotta fight that. And it's like, no, this is never getting easier. When, making life, when is the breakthrough going to come when this is going to get easier?
As time goes on, it becomes more difficult.
It is.
Right.
It's getting harder and harder. Yeah.
Well, back in Darwin's day, they just looked at the cell as what, a glob of jelly or nothing as complex as we know it is today. Actually, James Carter, again, in the chat, he's bringing up some really good points. He says, no polymers equals no life, since DNA, RNA, protein, and complex carbohydrates are all polymers that would hydrolyze, oxidize long before they would spontaneously grow to a functional length.
Yeah. Yeah. Absolutely. This is the other thing that's amazing that I love to do this demonstration for people. DNA, and RNA, these things just think about their size. Inside the nucleus, there's 6 feet of DNA. 6 feet of DNA. And wow. This thing is incredibly thin and tiny. And the fact that it's as robust as it is crazy. You kind of have to heat it to 90 degrees to denature it. I think, 90 degrees Celsius. And so there's some denaturing. So it's actually a pretty robust from a heat perspective. Now, from a radioactivity perspective, you can break it down, and that's what's happening. But just because it's robust doesn't mean it's robust in the way, like, how do you get it apart then so you can read the code? And it's all smushed up inside. And so I love to just take string and put it inside like one of those Easter eggs and then just see how much you can get. And like, well, this is really hard to get in here. Like, just the mechanics of putting DNA inside, and putting string inside of a cell is incredibly challenging. And so if you could build it inside of this tiny thing, well, that's even harder to do, is to build the string inside of a cell or inside of an Easter egg. Oh, just build the string inside. Then you don't have to worry about it. That's even harder. And so then you think about, this is amazing to me. You know how many miles of DNA are in your body? You guys have any clue how many miles?
No.
6 feet times, the estimates are kind of 40 trillion cells. We'll just kind of rough. 40 trillion times 6 feet. How far is that? From here to the moon? Here to the moon?
I could never run that far, put it that way.
You know how far is that? People have done the calculations. It's from the sun to Pluto and back 12 times.
Wow. That's amazing.
And if any of that goes wrong in your body, you got cancer and you're dead. You're done. You have that much DNA in your body right now. From the sun to Pluto. I'm not saying Pluto's a planet. It's just a place.
Right.
From the sun to Pluto, 12 times. That's how much DNA. And when that goes wrong, you're dead. Or you'll get cancer and you'll be dead sooner rather than later. And so if that's not being fixed, those polymers, you can make them, but you have to constantly be fixing them. Right. And then mechanically, how do you get them in the nucleus and then get them out to read so you can go take some of the code out there on the mechanics? Forget the chemistry, just the actual mechanical maneuvering to get it in and read it is incredible. It's incredible.
Yeah, to me, it's mind-boggling that anybody could believe that this has all happened, by chance for no reason at all. The things you're saying are just fascinating.
If we were, this is my new thing, my thought. If we stumbled on a planet and we saw a drill there, we saw a hammer, we saw, chemistry set, or you see things that you go, wait, that's a machine. There was somebody here.
No, it got there by chance.
Yeah. Oh, so I know there was somebody there, and yet now we're looking at the exact same stuff now. Oh, those enzymes, there's like little machines. Oh, that cell is like a little factory. That all happened. What? We wouldn't do that if we were looking for life on another planet. We go, man, look at that. Someone left a drill here. That planet that's a drill producing planet there. No.
Well, wasn't it Francis Crick? I believe it was him. He proposed directed panspermia based on the complexity, right? Based on the complexity. Yeah. So he was admitting design, but he's just attributing the design, obviously, to
absolutely. That's what we're seeing over and over again. Design is how you do it. Design is how you do it.
Well, we just got a good super chat in here that I should read. $10. Thanks so much. He says, Dr. Hayes, do your colleagues ever express frustration or dismay because of the ever increasing complexity discovered in biology?
Well, we try to ignore biology as much as we can because we're chemists, but no, absolutely, no, just joking. No, absolutely. We got our biochemists. We've got all of our work touches biology in some way or another. We have colleagues that maybe have studied active sites and enzymes, or doing some computational modeling. Where at some point it is about understanding how life works in various aspects, or making molecules to treat disease. We have to do that to understand how life works, and it's just we're baffled. It's like, what? Again, there's just the complexity that's there, and we're in awe. We're in awe. Just many chemists are quiet. I'm not.
Well, we love how outspoken and passionate you are. I mean, this has been a great interview, Dr. Hayes.
I blame my mom.
There you go.
My parents' fault. Yeah.
So we've got another good one here. Super chat from Doki. Doki says, a debater said the other day that an unlikely abiogenesis event just had to happen once, and then evolution can take over after that. Is this wishful thinking?
Oh, man. That is so wishful thinking. Right. Because that's the point. Okay, just think about it for a second, right? How often do things die around here? Right? Let's say you made life. Woohoo! It washes up on shore, and then boom, a rock rolls on it and it's crushed. No, you have to do it over and over and over and over. You need a factory that is turning out these little protocells, or something's going to kill it. A wind will come by, lightning or heat or cold. It'll be something is going to come along. And there's just so many ways to get destroyed on this planet that, that's a great point. You just have to keep making it. So that
and Dr. Hayes, aren't these protocells very unstable? They're breaking apart. They have to be protected in ways that are so unrealistic.
Yeah, absolutely. I mean, even our living cells, for most living organisms, our cells are pretty fragile. What about those thermophilic ones? Yeah, that's thermophilic. There's examples of things that are pretty robust, but in general, a cell is, they're kind of fragile things. And so the protocells, I think I don't know for sure, but my knowledge and understanding seems they're not any more robust. Yeah.
Well, it's so many good points. Again, in the chat, we've got a James Carter who's bringing up a lot of good points. People are loving this interview, Dr. Hayes. He's saying, actually, nucleic acids are very fragile. Cytosine is the least stable nucleobase with a half-life of I just lost it here. With 59.2 years at body temp in purified form in a sealed ample primordial RNA would never last.
That is great.
Is that Dr. Carter?
What's that?
Is that Dr. Carter from CMI?
No. This is James Carter. I'm not too sure he is, but he's bringing up lots of good points.
I know, it's just suspicious a little bit.
If you want to comment on that, Dr.
Those are some great points. I mean, the information I'm referring to is DNA, double-stranded DNA. If you're trying to get it to unravel and to do replication stuff in the lab, you have to heat it and you can unravel it. But the bases themselves that go in there, actually, I'm not really sure. So that's some really good information. I can just tell you from experience, organic molecules, which is what I did, which I built. I actually still have a company where we make nanomaterials. And this stuff just, anything is, if it's made out of carbons, it's made out of hydrogens and oxygens, they're fragile. Honestly, they're fragile. It's hard to make things that are not fragile out of those materials. You need metals. You need these framework materials, organic materials. Organic chemistry is my reference there. They're fragile. That's just the way they are.
It's amazing, and like you said earlier, even if you do give them everything that they need, and they've still got the information problem. So that's one problem.
Absolutely. You still got the information problem, and so I mean, if they want to put money into it, fine. But it's you're pouring money down a hole. And honestly, I should take it back. It's not fine, because this is money that could be spent on. Okay, how about fighting COVID or helping fight cancer And so you're kind of sucking away some of the money from there. So that does get me a little riled up that we're draining money off that could be helping people. So in a real quick, tangible way. And so maybe at this point, it should be funded by external funding mechanisms rather than government funding. We really should be funding helping save lives. Honestly.
Amen. I 100% agree. Another good question here. Super chat came in. Doki Doki Bible Club says, "Is it lazy science to say that that we're close enough in the lab? I guess to abiogenesis."
I would say it's completely dishonest science. You're making a mountain out of a molehill at that point. We are so far away that any respectable chemist looking at this information would go, no, we don't read headlines. We read the papers. And if you're going by the headlines, we've been there 12 times already, or 20 times, or 100 times. We've solved this problem. If I go by the headlines, we solved this problem, 20 years ago. But that's not what the papers are saying.
No. And you notice that with the articles, the headlines, they try, and people who don't look past that, they're going to see, life created in the lab, and then they're just going to automatically believe it. They're not going to dig deep.
Yeah. Scratch at the surface, ask somebody that does. Hey, I get asked all the time. Hey, what is that article? What's that video trying to say? And okay, let's go read it. And they didn't do very much. Oh, Dr. Carter by Loma Linda. Hello, Dr. Carter. I don't know if I know you, but I do know Loma Linda University. That's our sister school. Yay!
Small world. Well, thanks for being here, Dr. Carter. We've appreciated your questions, comments, super chats. Another question, interesting one from Sunflower in the chat says, "Do abiogenesis people claim that it only happened once? Or do they suppose it could have happened several times, but ultimately the cells or organisms were completely wiped out?"
Well, I mean, they hope that it happened once and then it would just, it took off from there. But I think the reality is, and when you look at a lot of the evolutionary thoughts that, hey, things had to happen multiple times. So you're really asking for the miracle, the lightning to strike, many times.
Right.
And that just makes it even more unplausible in my mind, where you've got to get them all those ingredients together again. Hey, let's call them ingredients. This is how people can relate to them, because we bring ingredients together to make food. And because chemicals, people don't really have a good sense of what chemicals are. They just think they're these mystical little good and bad things, and they make up stuff. But we know ingredients degrade. Ingredients help make things. But ingredients have to be made. You have to go make the ingredients. Even somebody had to make the ingredients. The chocolate chips they show up on my shelf, but someone had to make them.
Yeah, such a great point. Actually, we did have a question. I think we did kind of touch on this, but from our good friend John Maddox, who has helped with a lot of these interviews, he's asked a question. He said, "We hear all about how DNA synthesis is super easy and creating custom sequences is child's play. Can you explain a little bit how those machines work, and if what they do is even remotely similar to abiogenesis requirements, Dr. Hayes?"
No, nothing similar. A lot of these machines they've worked out the concentrations, they've worked out the time, they've worked out the temperature, they've worked out the purity of the nucleobases, and many of them have been designed, keyword, to have special linkers in them, so that under the correct conditions and concentrations, you bring in two ingredients and you put them together, and that helps to connect two of them. And then I think you then got to protect and deprotect or unprotect another functional group. Boom. Then you can bring in another nucleobase. And you have all of these very intricate steps of monitoring the time, temperature, and everything. And it's a machine that will do that. It's pretty tedious, but it's repetitive. And this is exactly what a machine can be built and designed to do, and as this repetitive work. But there's very carefully, and it doesn't produce very much. Honestly, these are like micrograms. I don't know if they're even micrograms. It may be less than that. They produce a little bit, but it's enough to do some interesting work with them. So it's machines controlling very well characterized steps that we know. So it's not a mystery how to bring these things together. We know how they come together, and we can program using our intelligence machines to do it for us.
Well, phenomenal answer, Dr. Hayes. It's been an honor to have you on. I know we've been going for about an hour and 10 minutes. I don't want to keep you all day.
Yeah, I love doing it. I mean, I don't know what, this just has triggered something in me in the last few years. And I try to incorporate it in my class, and it was just too much. There's just too much information that our students need to know to balance to balance everything. It's not even to tip the balance. I think it tips the balance, but just to have some honesty in this discussion, just to have some honesty in the discussion. Look, that's junk science. It's been disproved. Oh, it is? Really? Yeah. Well, I didn't know that. It was in our textbook. Okay. Well, that's why we gotta have some balance here, and because it's not balanced, they're not fair and balanced in the scientific world. This is the misunderstanding that which many scientists know once you get into there, that once you get into science and you start looking at it, it's not this pure industry that's led purely by hypothesis. They're following the money because they're an industry, and they're following, hypotheses and broken ideas. So anyway, I needed to get my PhD and to do research in this world to understand and to see that. And so, hey, they're humans. That's okay. We're all trying to make a living. I understand that. I get that. And we have our theories, but at some point we got to let the science speak, and let's see what the science is actually saying.
Amen. I could not agree more, Dr. Hayes. I love what you're doing. I love the passion. You're doing a great job. And I want to thank you for that. And Tony, if you had any final questions, final comments.
Yeah, just excellent. We'd love to have you back and some more discussions,
Absolutely. I'm all for it, you guys. I appreciate what you're doing. I need to do better. You guys are helping me, honestly, because I really want to get these thoughts and information out there. And I'm just busy teaching, and I go out to little camps and stuff. And I've got young kids, so I'm always doing stuff with kids. And they obviously, this is way above their intelligence level at this point. But yeah, anyway, curious of his thoughts on, oh man, this is good, on life on Venus.
Yeah.
No way. Well, partly I would go for a number of reasons why there's no water on Venus. We need to have water at least. And Venus is so hot that it's driven off all the water. And something interesting else about Venus, there's an electric field on Venus. This is surprising, really cool stuff. That they think the electric field is strong enough that it's split. If there was ever any water, it split the water and then shot out any oxygen and hydrogen from that, and ionized it and shot it out. NASA has a really cool video on the electric field of Venus. So possibility of life, no, it's way too hot, first of all. Life only exists at this kind of temperature range, the Goldilocks zone that Earth is in, and not a chance. I mean, finding life on Mars,
desperation at this point.
Only if we leave it there, buddy. That's only.
Yeah, exactly.
But Dr. Hayes, we can't even do it in the lab under hospitable, good environments. How are we going to push it to other planets? It seems like a great episode for Star Trek, but I mean, I just
no,
it doesn't seem realistic.
No, it's hard. And so this is where it would be fun to come back and talk about what are the conditions to have even water on a planet. This is all ignored. No one's thought. Why is there nitrogen in our atmosphere? And so the people just ignore all of this stuff. And if we just start scratching at the chemistry a little bit, we're like, oh wow, whoa, did somebody design our planet? No, it can't be, because there's billions of suns. There's billions of planets out there. There have to be tons of these little Earths out here somewhere. I don't know. I'd love to give you guys some more thoughts on that.
I would love to have you on and discuss that. You've, like I said, the audience is loving it. We've had lots of good questions, lots of good input. And like you said, it just seems like, and all we're asking is for honest science, honest discussion.
Yeah, exactly.
Well, even here, James Carter, Dr. Carter says it, another good comment. The law of diffusion is why chemists do experiments in test tubes. And not
I don't know. We did some chemistry experiments in our pool this summer, but shame on those kids. Anyway.
Yeah.
No, that's exactly right. You have to do that. You have to control diffusion. Yeah, we didn't even talk about diffusion. Oh my word. Yeah, there's one thing after the next from a chemical perspective, which is really hard. For a lot of biologists to work at, because we spend a lot of time in our chemistry classes thinking about the conditions for doing analytical work, for making things. And it just takes a lot of working chemists many, many years to think about all these conditions. Yeah.
Well, it's problem after problem. And Tony and I always talk about this. We could probably discuss these things all day. Yeah. There's always it's too much fun, and it's so important. You know, it is. It's
very important. So you guys are doing a good work here, and hopefully more and more people will start listening. And but people. And I got into my own family. They don't really care. They hear this. Most of my family loves what we're doing here. I get my family involved. My kids help do the demonstrations chemically to help prove how nature works, what is natural and what's unnatural. Yeah. Phosphine and I got to talk to this James Carter. I'm going to hang down.
He'd be a good addition. Well, yeah, no, it's like I said, it's important topics. This has been an awesome interview. We've had a lot of good interaction in the chat. We would love to have you on. Like I said, we love your passion, your enthusiasm, and of course your knowledge. So, yeah. So, any final words at all, Dr. Hayes?
Honestly, if people just go read some more, be open-minded, but be open-minded, and let's see what all the science is saying. And you kind of have to dig around a little bit, because there is manipulation of the scientific information that does make it out there. There's not a fund. We do not, those who are looking at origin of life, who may have a different view of how it started, they're not funded, and you're going to have a hard time seeing that stuff in the journals, and then you won't see that in the news reports. So you're not going to get that from mainstream places. And so I think it's important to know that. But, hey, we have YouTube. We have other places that we can get the message out.
Exactly. And I think that's the most important part is getting the message out there, because it sounds like a lot of students, and I think it's a blessing when students have you as a teacher and can learn about these things. But it seems like 95% of students won't ever hear about this. They won't ever hear about the major issues with origin of life.
Yeah, not even on our Christian campuses, honestly. Which is a shame. So I hope to at least try to change some of that.
Well, that's our goal is to get the message out there, stand for the truth. And we would, as many people are saying in the chat, we'd love to have you on again, Dr. Hayes. And it's been an honor. It's been a blessing.
Thank you. I'm honored. And let's make it happen.
Awesome. Okay. Well, I want to thank the audience for being so lively and giving us a lot of good input, questions, super chats. And before I shut it down, any final words, Tony?
Thank you, man. Thanks, Dr. Hayes. God bless you, brother. We'll see you again. Appreciate it.
God bless you guys. Thank you very much.
Awesome. God bless everybody. God bless.